Hi, today we are going to discuss something about nucleotides. Nucleotides are phosphoric esters of nucleosides which are having 5' phosphate at carboxyl group at carboxyl end, and hence they are called nucleoside 5-phosphate. So the sugars which are carrying an deoxyribonucleotide or ribonucleotide, if the pentose sugar is ribose, then we can say it as ribonucleic acid, and if the pentose sugar is deoxyribose, we can say it as deoxyribonucleic acid. What are the functions of nucleotide? Nucleotide can help in formation of polynucleotides. They can also generate certain compounds which are metabolically active compounds, and they can participate in various metabolic processes. And what other things they can do? They can form coenzyme. They can participate in formation of coenzymes like NADP plus, NADP plus, and coenzyme A. They can also participate in formation of energy currency like AMP, ATP, GTP, etc. And they can also form secondary messengers like cyclic CMP and cyclic GMP. So if we are discussing about cyclic AMP and cyclic GMP, we can also discuss about ATP, something about ATP. So what is ATP? As it is well known and very important topic to be discussed. The ATP is also known as adenosine triphosphate. It is also energy currency of the cell, very important role it plays in our cell, that is formation of energy currency or acting as a energy currency of cell. It also act as donor of phosphate or donor of pyrophosphate inside the cell. The other thing which we can discuss beside the structures of nucleotide is polynucleotide. Polynucleotides are formed by nucleotides only. Uh, the polynucleotide are formed by condensation reaction. The condensation reaction occurs between the alcohol of 5' phosphate of one nucleotide to 3' hydroxyl group of the other one, with the elimination of one water molecule. The linkage between the acidic phosphate or at physiological pH of nucleotide, it is also known as phosphodiester bond. So the linkage between the two nucleotide is also known as phosphodiester bond, and at the pH, physiological pH, they exist in polyanionic condition. So they are also existing as polyanions because the charge contained by them is negative because of presence of phosphate group. All nucleotides are formed in the direction of 5' to 3'. So they are all having directionality. They are all having a certain path which they follow in joining while joining together. So all nucleotide exhibit 5' to 3' directionality, that is the linear structure, that is in the formation of linear structure of nucleotide proceeds in 5' to 3' direction. The order in which nucleotide occur in polynucleotides describe its primary structure. The sequence of nucleotide sequence in nucleic acid is written from left to right, from 5' to 3' end. So 5' is taken on the left side and 3' end is taken on the right side. The interchain and intrachain pairing of complementary nucleotide gives rise to secondary structure, and the 3D arrangement of nucleic acid, that arrangement of molecule in space, is also referred to as its tertiary structure. Rest the structure in detail. We will discuss about that in the upcoming videos. So kindly